Hello everyone, welcome to my YouTube channel Indian Anatomist. In this video, we'll discuss about the histology of bone. What is bone tissue? Bone is a special type of connective tissue composed of cells and matrix that is made of fibers and minerals. The mineral deposition within the bone matrix resulting from the process of calcification contributes to its fundamental hardness. As a result of mineralization or calcification, bones exhibit increased load-bearing capacity, thereby functioning as a rigid skeletal framework for the human body, and also serves as attachment sites for muscles and organs. Furthermore, the bone undergoes hematopoiesis, which refers to the process of blood cell formation, and plays a vital role as a store for essential minerals such as calcium, phosphate, and others. Approximately 99% of calcium in the body is stored in the bones, which serves as a primary source of body's daily calcium intake. Prior to exploring into the field of histology of bone, it is essential to obtain a comprehensive understanding of the bone's inclusions. Bone includes cells and matrix. The cells of bone comprises osteoblasts, osteocytes, and osteoclasts, whereas the matrix includes the organic and inorganic substances. Now coming to the cellular components of matrix. Number 1 is osteoblast. These are the cells that are responsible for the synthesis and deposition of new bone matrix. Osteoblasts are located on the both periosteum and endosteum layer of the bone, and these have a vital function in the process of bone formation and regeneration. The second cellular component of the bone is osteocytes. Osteocytes refer to fully developed bone cells that are situated within the bone matrix, specifically within the minute cavities known as lacunae. In this picture, we can see the arrow-marked structures. These are the lacunae within which the osteocytes are lodged. Osteocytes are responsible for preserving the bone matrix, detecting mechanical stress, and engaging the bone in remodeling. The last type of bone cell is osteoclast. Osteoclasts are monocyte fusion cells, which is also called as bone-eating cells. These are characterized by their huge size and multinucleated nature. Osteoclasts play a crucial role in the process of bone resorption, which involves the breakdown of bone tissues. Osteoclasts produce enzymes and acids, enabling the process of bone tissue remodeling and repair. This facilitates the breakdown of mineralized matrix and hence allowing for the occurrence of new bone formation. Now, let us see about the matrix. The bone matrix is predominantly composed of osteoid, the immature bone matrix, which comprises both inorganic and organic constituents. Coming to the organic components of matrix. Number 1 is collagen fibers. Predominantly, these collagen fibers are composed of type 1 collagen, which offers flexibility and tensile strength to the bone by forming a triple helix structure, thereby enhancing its ability to withstand stretching forces. The second organic component of matrix is proteaceous matrix, which comprises a range of proteins including osteocalcin, osteonectin, and osteopontin. Proteins associated with mineralization, cell signaling, and bone remodeling are of utmost importance. Next, we will discuss about the inorganic components of bone. The first inorganic compound is hydroxyapatite. These are mineral crystalline compound that is predominantly made of calcium and phosphate ions. The deposition of these crystals occurs within the collagen matrix, thereby imparting compressive strength and the bone with stiffness or hardness. The other inorganic components of the matrix includes magnesium, sodium, potassium and carbonate ions, in addition to calcium and phosphate within the bone matrix. These ions have a significant role in the processes of mineralization and the physical characteristics of the bone tissue. Dear students, so far in this video, we have covered the basics of the bone, including its organic and inorganic components as well as its cells and matrix. Now I shall proceed to explain the histological characteristics of compact and spongy bone. Histologically, the bone tissue can be classified into two types. Number 1, compact bone or cortical bone. Number 2, spongy or cancellous or trabecular bone. Most of the medical colleges in the country do not provide the histology demonstrations or examinations of spongy bone. Acquiring the knowledge is beneficial as it allows for exploration of theoretical concepts. First, we shall examine the concept of compact bone. Compact or cortical bone. The diaphysis of long bone is formed by this compact bone. It gives the bone strength, stability and protection, and it looks solid to the naked eye. The histology of compact or cortical bone can be studied in two ways: the transverse and longitudinal sections. The matrix is organized into thin layers known as lamellae when observed under the microscope. The lamellae can be classified into two types. 1. Outer circumferential lamella, which is located at the periosteum. Number 2. Inner circumferential lamella, which is located near the endosteum, that is close to the bone marrow canal. A third variety of lamellae can be observed in these sections, that is the concentric lamella. Between the circumferential lamellae, that is outer and inner circumferential lamellae, a wide area of lamellae are arranged in numerous concentric pattern, which is called as concentric lamellae. The concentric lamellae is otherwise also called as an osteon or the Haversian system. Is a structural and functional unit of the compact bone. What is a osteon or Haversian system? Osteons or Haversian systems are cylindrical structures that are aligned parallelly to the long axis of the bone. Bone lamellae are comprised of osteocytes that are located within the lacunae, which are almond-shaped gaps. Every lacunae exhibits a network of small canals known as canaliculi. These canaliculi serve to connect the neighboring lacunae or the Haversian canal. The communications between the osteocytes occur via the canaliculi, which serves as a conduit for obtaining blood and nerve supply from the central Haversian canal. What are interstitial lamellae? These are uneven patches between the osteons, which are called as interstitial lamellae, which are nothing but the erosion or remodeling remnants of the bone. What is a cement line? The cement line is a refractive line between adjacent osteons caused by matrix alteration. Oakman's canal. Haversian canals are often joined by transverse Oakman's canal or perforating canals. The Haversian canal contains the neurovascular bundle. In this picture, we can see both the transverse and longitudinal sections. In longitudinal section, we can see the Oakman's canal and a Haversian canal. Thereby, the blood vessels are distributed from one part to the other part of the bone tissue. Dear students, as of right now, I have discussed the transverse section of the compact bone. Dear students, as of right now, I have discussed the transverse section of compact bone. If you find concentric lamellae, then it is a transverse section of bone. What does the histology of compact bone look like when it is examined in longitudinal section? It's very simple. Longitudinal section of compact bone. Haversian canals are often joined by transverse Oakman canal or perforating canal. No osteon or Haversian system or concentric lamellar pattern are seen in longitudinal section of compact bone. Like transverse section of compact bone, in longitudinal section also we can find the lacunae and canaliculi. In this picture, we can see the Haversian canals which are vertically oriented and the Oakman's canal which are horizontally oriented. As we have discussed earlier, the second type of classification of bone is the spongy or cancellous or trabecular bone. Spongy bone is composed of lattice-like and porous components and is located within the flat bones and at the ends of long bones. By nature, the spongy bone has a porous or trabecular appearance. Spongy bone is distinguished histologically by the presence of network of trabeculae, which are thin bony plates or rods arranged along the stress lines. In this picture, the arrows represents the trabeculae. In this picture, we can see the green dots which represents the bone marrow. The bone marrow fills the empty spaces between the trabeculae. Unlike the compact bone, spongy bone lacks osteons, while still possess the osteocytes which are distributed within the lacunae and are connected by canaliculi. Now, we will discuss about the functions of compact and spongy bones. The compact bone maintains structure, resists bending and torsional pressures, and protects organs and bone marrow. Calcium and phosphate are stored in compact bone. The functions of spongy or cancellous bone. The spongy bone material offers both structural reinforcement and a lightweight nature. The porous nature of its structure facilitates the movement of nutrients and waste substances to and from the bone marrow. Moreover, spongy bone is involved in the process of red blood cell synthesis within the bone marrow. The functions of compact and spongy bone together. The functions of these two distinct forms of bone tissue are complementary to one another. Compact bone is responsible for providing strength and provides protection, whereas the spongy bone allows for the interchange of nutrients and contains the bone marrow, which is essential for hematopoiesis or blood cell formation and also the fat storage. Dear students, with this above said knowledge of compact and spongy bone, now we will discuss how the compact and spongy bone looks alike in microscope. Many compact bone slides of transverse and longitudinal sections in histology lab are unstained. Small bone fragments are rubbed across hard surfaces to create thin plates of bone. After washing, it is placed on the slide for observation. Because of this, the osteocytes are washed away and leaves a small spaces called lacunae. Students use only HP pencils to draw compact bone diagrams in their record notebooks. Dear students, let's talk about the horizontal and longitudinal sections of the compact bone from a histological or microscopic point of view. First, we will look at the transverse section of compact bone. This slide displays the transverse section from low to higher magnification, which shows several concentric bone lamellae, also known as the Haversian system or the osteons, along with interstitial lamellae that lie in between them. When you zoom in on the slide, try to focus on one or two osteons to study. While we are still zooming in, each osteon has many concentric rings that circle the Haversian canal. While we are still zooming in, each osteon has many concentric rings that circle the central Haversian canal. Close by, there is a white refractive line called the cement line, which are present between the two osteons. At a higher magnification, each Haversian canal is surrounded by many empty spaces called lacunae, which are home to single osteocytes. The osteocytes are washed away because the bone tissue is grounded, leaving gaps or lacunae. Each bone lacuna is connected to neighboring lacunae or to the Haversian canal through small canals called bone canaliculi. Now, we will examine the longitudinal section of compact bone. It is widely known that there is no concentric lamellar pattern in the bone portion of longitudinal section. In this part, all the Haversian canals are arranged in parallel manner, and these canals are joined horizontally by perforating or Oakman's canals. The area surrounding the canals has empty gaps known as lacunae, which provide the impression of small canals called canaliculi. The canaliculi serve as conduits for the protoplasmic processes of osteocytes, allowing communication between nearby osteocytes and a direct contact with the Haversian canal for the exchange of nutrients and disposal of waste products. Now, let us see about the microscopic structure of spongy bone. This is a slide from the epiphysis of long bone showing the spongy part, which is stained by hematoxylin and eosin. From a microscopical point of view, the spongy bone is made up of trabeculae, which are thin strips of bone that connect to each other and surround the bone marrow and blood vessels. It makes up the core of the bones, which is lighter and full of marrow. The spongy bone has less tightly packed trabeculae, which make up the bone lighter when compared to the compact bone, which makes it heavier. The trabecular network increases the amount of room in the bone, which makes it lighter, and this arrangement also reinforces the bone, which makes it strong enough to bear the weight. There are no specific lamellar patterns which can be observed in the spongy or trabecular bones.
I hope all of you would have understood in detail, which will be helpful for your studies. Let us be connected for the upcoming videos. Thank you all.